Started?
Yes, sir.
Thank you. Okay, let us get started. Dr. Subhasis Pal, a CDRA alumnus, is here today for the webinar. Subhasis is currently a postdoc at Emory University in Atlanta, Georgia, USA. Subhasis got his PhD degree in 2019 and immediately moved to Emory University to work in the laboratory of Professor Roberto Pacifici, a pioneer in the field of osteoimmunology. Subhasis happened to be a prolific researcher, as evidenced by his CV, having published 42 papers in prestigious journals, including Biomaterials, ACS, Applied Materials and Interface, Bone, Calcified Tissue International, Journal of Ethnopharmacology, and others during his PhD. He is also the co-inventor of a patent which has a US patent, approved US patent and licensed to an industry. In 2019, he was awarded the coveted CDRI Best Thesis Prize in recognition of his scientific achievement. He is still as productive in his postdoctoral position as he was during his PhD. Subhasis has already authored 3 publications in JCI, Journal of Clinical Investigation, during his 3 years as a postdoc. And he is currently editing a special issue on perspective on bone research for frontiers in endocrinology. So today we will hear about his recent research on titled Gut Microbiome's Role in Melanoma Bone Tumor Formation. Welcome, Subhasis.
Yeah, so thanks for the nice introduction. Good evening, all of you. It's an honor for me to present my postdoctoral work in front of my old lab members and colleagues. My name is Subhasis, and I completed my PhD from CDRI in 2018. After that, I joined Emory University as a postdoctoral researcher. And today I am going to present my postdoctoral work, which I have been recently published in JCI journal. Title of my presentation is Gut Microbiome Mediated Regulation of Melanoma Bone Tumor Growth. Now, all of you may know that human body is inhabited by millions of microorganisms, which all together are known as human microbiota. They are found on skin, nose, mouth, and especially in the gut. The human microbiota is involved in healthy growth and protecting of body from invaders and helping digestion and also benefit and support our immune system. So they are absolutely essential for our survival. One interesting fact is that we have more microbial cells in our body than the number of total human cells present in our body. Approximately, the microbial cell present on surface and within our body is 10 times higher than the total number of human cells. And among all these microbiome population present in our body, gut microbiome is the largest, richest, and most complex microbiome within our body. More than 1,000 different types of bacteria exist in the human gut. Now, human evolves an intimate symbiotic relationship with gut microbiome. To be healthy, we need to take care of beneficial intestinal bacteria by eating healthy food. Studies in human and animal models have implicated that gut microbiome is a regulator of bone mineral density, postnatal skeletal development, bone tissue material property maintenance, and bone mineral absorption, and also regulate the pathogenesis of osteoporosis. Now, if you go to the recent studies, and they'll currently link gut dysbiosis with the cancer progression also. And recent studies also showed that gut dysbiosis regulate the response of cancer therapeutics. However, little information is available on whether the gut microbiome play a role in osteolytic bone lesions and how immune system regulate tumor growth under the influence of gut microbiome. So, in this study, we focused on malignant melanoma, a frequent and aggressive form of skin cancer. Our primary question for this study is, does gut microbiome regulate the establishment of growth of melanoma bone tumor, and what is the possible mechanism? Now, to investigate the extent to which gut microbiota influence bone tumor growth, we performed an intratumoral injection model, and we used luciferase expressing B16 F10 cell lines, and we inject these cell lines into 12-week-old C57 black 6 wild type mice. To ablate the gut microbiome, mice were treated with broad spectrum antibiotics composed of ampicillin, vancomycin, neomycin, and metronidazole, administered via drinking water, and the whole duration of the antibiotic treatment is 4 weeks, starting 2 weeks before the tumor injection, and also we maintain the antibiotic treatment throughout the tumor progression, and we monitored the tumor growth by using IVIS scanning. Now let's come to the result. What we found: after B16 F10 melanoma cell line injection, tumor growth was monitored at day 5, day 10, and day 15. IVIS quantification data showed that gut microbiome depletion, like this group is the gut microbiome depleted mice, and this group belongs to the control group. And you can clearly see that gut microbiome depletion triggered the tumor growth, and we observe a peak tumor growth at day 15. Although at all the time point like day 5, day 10, and day 15, the tumor growth is higher in gut microbiome depleted group compared to the control group. Now, to evaluate tumor-induced perforation and lesion, we perform microscopy analysis of the tumor-bearing bone. In this slide, you can see the representative image of tumor-bearing tibia. Yellow color is showing perforation, and red color is showing ectopic bone growth. It is clearly visible from this image that the antibiotic treated group have clearly higher number of red color and yellow color compared to the control group. And we also found similar trend when we do the microcity analysis quantification. We performed the analysis at day 15, and we measured cortical perforation and ectopic bone growth in distal metaphysis and diaphysis area. We found that the bone lesions were greater in tumor-injected bones of gut microbiome depleted mice compared to the tumor-injected leg of control mice. To confirm that the effect of broad-spectrum antibiotics on bone tumor burden and osteolysis arise from direct effect of antibiotic on intestinal bacteria and not from any off-target effect of antibiotics, we performed a second experiment where we used a set of non-absorbable antibiotics, a mixture of neomycin and bacitracin, dissolved in drinking water for the whole duration of experiment, and we start the antibiotic treatment 2 weeks prior the tumor cell injection. And then we inject the tumor cells, and we keep the mice for 15 days. And at the end point, we do microcity, and throughout the tumor progression time point, we do intermittent IVs. So our data with non-absorbable antibiotics showed that local tumor growth peaked at day 15, and at each time point, tumor bone burden was greater in non-absorbable antibiotic treated mice compared to its untreated control. We also quantified the data, and our quantification also a similar trend. Like here you can see 5-day, 10-day, and at day 15, in gut microbiome depleted mice, the tumor burden is higher. Moving to the next slide. So I guess I'm back online again, right? Hello.
Yes, we can hear you. Please reload
Uh,
your, yeah, please share your
yeah,
slides again. And you are
uh,
showing the, yeah, you are showing the slide on the non-absorbable antibiotic and bone effect.
yeah, non-absorbable antibiotic and like, did I go through the IV's data
Yeah,
and
I this
the
data, that
micro?
is where that is where it froze. Yeah, that is where it froze. Yeah.
Okay. So our data with uh, non-absorbable antibiotic treatment showed that local tumor growth peaked at day 15, and at each time point, tumor burden was greater in non-absorbable antibiotic treated mice compared to the control mice. Our IV's quantification data also show a similar trend. Next, we performed microcity analysis of the tibia harvested at day 15, and we measured the bone perforation and ectopic bone growth, perforation indices, and ectopic bone volume indices showed that gut microbiome depletion by using a non-absorbable antibiotic increased the bone lesions in this group compared to the control group. Since non-absorbable antibiotic and broad-spectrum antibiotic were both effective in altering the measured indices and promoting tumor growth, it is unlikely that the activity of antibiotic is due to an off-target effect of these agents. Gut microbiome depletion is solely responsible for the increased tumor growth. Now, to evaluate the involvement of immune system in this process, we measured a complete profile of different immune cells like CD8 positive T cells, cytotoxic CD8 T cells, NK cells, T helper cells, Tregs, and MDSCs. Although gut microbiome depletion reduced several cell population in the gut, but it didn't affect the absolute frequency of CD8 positive T cells, cytotoxic CD8 positive T cells, Tregs, and MDSCs. I'm not showing those negative data in this slide. I'm only showing two positive data, like with NK cells and Th1 cells, which population we found that has been regulated by gut microbiome. So in contrast, frequency of both NK and Th1 cells increased in response to tumor and gut microbiome depletion and lowered the frequency of NK and Th1 cells at the tumor site. So in the gut, you can see that when we deplete the gut microbiome, the Th1 cell population and NK cell population goes down, and same happens in the bone marrow. Here you can see with NK cells and with Th1 cells, and with tumor challenge in the control group, you can see NK cells are going up and also the Th1 cells. So from this experiment, we identified that these two cell population is played the most important role and mostly is regulated by gut microbiome under the influence of tumor. So since intestinal immune cells are known to migrate to distant organs, we hypothesize that bone tumor growth may cause the homing of these immune cells from gut to the tumor site and thus regulating the tumor growth. And this may cause by a direct influence of tumor cells, or maybe like tumor cells regulating the bone marrow cells to secrete some cytokines and then regulate the effect. So both could be the possibility. To directly investigate the effect of cancer bone growth and NK and Th1 cell trafficking, we use this CADA mice experiment that we have already established in our lab and also used in our previous publication. These strains offer a sensitive means of tracking cells and cellular migration from the gut to the anatomically distant site of any leukocyte population, or basically you can track any cell. CADA mice ubiquitously express the photoconvertible protein CADA, which permanently changes its fluorescence emission from green to red upon photoconversion with near UV light. Once photoconverted in the intestine, red fluorescent cells can be detected by flow cytometer in other organ. For our case, our target organ is the tumor site. And we also know that photoconversion has no effect on cellular function and on the homing capacity of these cells. Now, in our case, what we have done is intratumoral injection of B16F10 cells were performed in 12-week-old CADA mice 9 days after the bone tumor were when actually we do a IV scanning and we found that kind of the 10th day is the suitable time point when the tumor growth is well established. So we consider like 1 day before 9 day for the laparotomy experiment. So 9 day later, when bone tumor was well established and rapidly growing, all animals were subjected to surgical laparotomy, and we exposed 4 pairs patch per mouse and photoconvert them by exposing to the UV lights for 2 minutes. And to ensure that no other cell were photoconverted, the whole mouse were covered with an aluminum foil blanket. Mice were sacrificed 24 or 48 hours post laparotomy, and the number of KIDA red NK cells and Th1 cells in pairs patch and in bone marrow was measured by flow cytometry. And this is our result. Our results showed that a decrease in the frequency of pairs patch KIDA red NK and Th1 cells at 24 and 48 hours. Injection of tumor cells markedly increased the relative and absolute frequency of bone marrow NK and Th1 cells at 24 and 48 hours. Like you can see, the NK cell percentage goes up here at 24 and it also maintained in 48, and the Th1 cell population also goes up. One thing you need to remember, all the cell quantification data is like red cells. That means we photoconvert these red cells in the intestine. So whatever cell we are detecting at the tumor site is coming from the intestinal area. Together, these data demonstrate that intraosseous melanoma tumor growth promote the migration of intestinal NK and Th1 cells from intestinal tissue to the bone. Now, to determine whether the migration of immune cells from the gut to the tumor site is microbiota dependent or not, again the same CADA mice were subjected to intratumoral injection of B16F10 cells with or without broad spectrum antibiotic treatment. 9 days after the tumor cell injection, all animals were subjected to laparotomy and pairs patch were photoconverted. Mice were sacrificed 24 hours later, and the number of KIDA red NK and Th1 cells in pairs patch and tumor site were measured by flow cytometry. Now we found that gut microbiome depletion prevents the increase in the frequency of these cells at the tumor site. You can see here, this is the control mice group, and this is the tumor bearing group. So in the tumor bearing group, the NK cells in gut microbiome intact mice goes up, whereas in gut microbiome depleted group, the NK cell population didn't go up. And same thing happens with the Th1 cell population. So we found that gut microbiome depletion prevented the increase in the frequency of these cells at the tumor site, demonstrating that melanoma induces increase in the migration of pairs patch NK and Th1 cells is microbiota dependent. We also verified that the cell migration was site specific, and for that we measured the red NK and Th1 cell population and at different organ like tumor site, liver, spleen in both control and tumor group, and we found that these red cells didn't increase significantly at other organs like liver and spleen. It only increased in the targeted way at the tumor site. And our necroscopy analysis during sacrifice also confirmed that liver and spleen tissues didn't have any tumor growth. So from this experiment, we can say that our cell migration is completely happen in a targeted way. Now, we confirm that the cells migrate from gut to the tumor site. Next thing is we need to identify the possible mechanism by which these cells are exiting from the gut and the possible mechanism by which the cells are entering into the tumor site. Now, the chemokine receptor SIPR1 and R5 are expressed by T cells, NK cells, and it's a well-known fact. These receptors promote the egress of intestinal T cell and NK cells in response to the sensing circulating SIP. To investigate the hypothesis that melanoma growth in bone promote the egress of Th1 and NK cells from the intestine through SIPR1 and R5 mediated mechanism, animals were injected with B16F10 cells and treated with SIPR1 functional agonist FTY720 and SIPR5 functional agonist BAF312. Now, this is a representation of our study design. FTY720 was administered by drinking water with a pretreatment of 7 days and maintained throughout the tumor growth progression, and tumor growth was monitored by IV scanning and microCT analysis. Our flow cytometric analysis showed that FTY720 had no effect on the frequency of pairs patch, NK, and Th1 cell population in either control or tumor injected mice. That means this drug didn't alter the gut population of these cells. Moreover, FTY720 did not affect the increase in bone marrow NK cells induced by cancer growth, as you can see from these two groups, while it prevented the tumor-induced increase of Th1 cells. This finding showed that the egress of Th1 cells from the gut to the tumor site is regulated via a SIPR1 mediated mechanism. To evaluate the effect of FTY720 on tumor growth, we performed IVIS analysis, and our result showed that the blocking of Th1 cells promoting tumor growth at all the time points with a maximum growth at day 15. Our quantification data also show similar trend. It's significant at all the time points. Next, we performed microCT analysis, which also confirmed that blocking of Th1 cells by FTY720 treatment promote bone perforation and lesions. Together from this experiment, we established that the physiological relevance of Th1 cell migration to the tumor site and Th1 cells present at the tumor site is important to regulate the tumor growth. Now, for second inhibitor experiment, we used BAF312, and it's a SIPR5 inhibitor. BAF312 was administered by oral dosing with a pretreatment of 7 days and maintained throughout the tumor progression time, and tumor growth was monitored by IV scanning and microCT analysis. Now, flow cytometric analysis showed that BAF312 had no effect on the frequency of pairs patch NK and Th1 cells in either control or tumor injected mice. That means this agent didn't alter the gut population of these cells. Moreover, BAF312 did not affect the increase of bone marrow Th1 cells induced by cancer growth. As you can see, the Th1 cell goes up in presence of BAF312. But this reagent prevents the tumor-induced increase of NK cells. From this finding, we can say that the egress of NK cells from gut to the tumor site is regulated via a SIPR5 mediated mechanism. Now, to evaluate the effect of BAF312 on tumor growth, we again performed IVIS analysis, and our results showed that blocking of NK cells by BAF312 promote tumor growth at all the time points with a maximum growth at day 15. Our quantification data also show the similar trend. Next, we perform the microCT analysis, and microCT analysis also confirmed that blocking of NK cells by BAF312 promote bone perforation and lesions. Together from this experiment, we established the physiological relevance of NK cell migration to the tumor site. Now, we proved, or we actually found the possible mechanism through which these cells are exiting from the gut. Now the turn is how these cells are recruiting to the tumor site. While the egress of Th1 and NK cells from intestinal lymphoid tissue and their entrance into the bloodstream is driven by SIPR1 and R5 and their ligand SIP. Based on current literature study, we hypothesize that CXCR3 CXCL9-10 axis could regulate the infiltration of Th1 and NK cells to the tumor site. This is well established that CXCR3 receptors are expressed by T cells and NK cells. The ligand CXCL9-10 expression can be induced by IFN gamma, which is mainly secreted by Th1 cells. NK cells also secrete IFN gamma, and the melanoma cells. And the tumor cells secrete largely these ligands. So this axis could play a major role in this Th1 and NK cell recruitment to the tumor site. Now, to check our hypothesis, we evaluate the expression of all the ligands CXCL9, 10, 11 at the tumor site. And our result showed that the expression of this ligand increased at the tumor site, and depletion of gut microbiome didn't alter the expression, suggesting that this is a tumor-driven ligand expression. So as we confirm the presence of this ligand at the tumor site, next we use CXCR3 knockout mice to evaluate the involvement of receptor. Flow cytometry analysis showed that the frequency of bone marrow NK cells and Th1 cells at the tumor site were increased by bone tumor growth in wild type mice. You can see, like in wild type mice, there is a significant increase compared to the no tumor control. And in CXCR3 knockout mice, this cells population didn't go up. You can see it's significantly down. This finding establish the critical role of CXCR3 receptor in NK and Th1 cells homing to the tumor site. That means if the CXCR3 receptor is not present to NK and Th1 cells, or like in other bone tissues, these cells recruitment won't happen to the tumor site. Establish the functional relevance of CXCR3 receptor mediated effect. We performed IV scanning by using wild type and CXCR3 knockout mice, and we found that CXCR3 knockout mice have higher tumor growth compared to the wild type mice. Our quantification data also showed the similar trend, and we found a highest tumor volume at day 15. Now, we also done a microcity analysis. CXCR3 knockout mice have more severe cortical perforation indices and ectopic bone growth. And here you can clearly see that the ectopic bone growth is like clearly very high, and it's very interesting pattern compared to the wild type mice. So that means the tumor burden is clearly higher in CXCR3 knockout group. From this data, we provide evidence that CXCR3 functions in the influx of Th1 and NK cells into the tumor site and impact bone growth. Now it's time to investigate the possible ligand. So to investigate the role of CXCR3 ligand, intratumoral injection of B16F10 cells were carried out in 12-week-old wild type mice, and then we treat these mice with anti-CXCL3 or CXCL9 neutralizing antibody and control antibody, isotype matched irrelevant antibody. Our results showed that the frequency of bone marrow NK cells and Th1 cells were increased by bone tumor growth in control mice, and when we treat these mice with CXCL9 neutralizing antibody, these cells population didn't go up. So that means this presence of this ligand is required for the recruitment of these cells into the tumor site. To establish the functional relevance of CXCL9 ligand, we measured tumor growth by using IVs. And here you can see that CXCL9 neutralizing antibody treated mice have higher tumor burden compared to the control mice. And our quantification data also proved this finding. We also performed a microcity analysis, and which showed that CXCL9 neutralizing antibody treated mice have severe cortical perforation and ectopic bone growth. Together, these data provide evidence that CXCL9 functions in the influx of Th1 and NK cells into the tumor site and impact tumor growth. So this is the summary of our whole study. Together, our study showed that gut microbiome restrain the progression of melanoma bone lesions in mice by promoting the expansion of intestinal NK and Th1 cells and enhancing their migration to the tumor-bearing bones. The egress of NK and Th1 cells from the gut and their entrance into the bloodstream are driven by SIP-R1, R5 receptor. We also found that the influx of NK and Th1 cells to the tumor site is dependent on the chemokine receptor CXCR3 and its ligand CXCL9. So this study reveals mechanism for microbiota-mediated gut-bone crosstalk in mouse melanoma tumor model. The observation that microbiome depletion accelerates bone tumor growth indicates that antibiotic-treated treatment and other cause of gut dysbiosis may have adverse consequence in melanoma patient. We recently published this study in the Journal of Clinical Investigation. This whole work is part of my postdoctoral project and result of 3 years hard work. I hope all of you found this study meaningful and interesting. I want to acknowledge NIH and USA Department of Health Service for providing funding, my mentor, Dr. Roberto Pacifici, for his guidance and support, and our whole team member, Dr. Dan Perrin, Tetsuo Yamamoto, Dr. Jones, John, and Dr. Wiseman for their constant support. And I found this picture suitable for the last slide. It's clicked by me at Death Valley, California. It's showing a postdoctoral researcher journey. It's very much similar, like keep walking in search of a suitable destination. Thank you.
Wonderful. It was a elegant piece of work and has been presented very lucidly. You have walked through your each data very lucidly, and it is I'm sure that people will have, the audience had no problem in understanding the data, very clean data, and I congratulate you on your this discovery, your entire team.
Thank you.
I particularly like the Keda mice experiment. It was
Yes.
a really very elegant experiment done. One thing you were talking about, SIP means this is sphingosine 1-phosphate receptors, right?
Yes.
Sphingosine 1-phosphate. Now, this SIP sphingosine 1-phosphate is quite extensively been implicated in many osteogenesis related processes, including stem cell recruitment and subsequent differentiation and survival of osteoblast, and even to bone marrow angiogenesis.
Yes.
Means the blood vessel vasculature system in bone. Now, when you are giving any inhibitor, how are you going to affect its roles, SIP's role in bone?
So if you are giving an inhibitor like we have seen in our previous experiment, if you treat the mice with FTY 720, then the direct effect is osteoblast activity actually goes down or suppressed. Like in fracture condition, if you treat the mice with FTY 720, then fracture healing got delayed. So definitely these inhibitors actually suppress the activity. But in our case, like we are only focusing on the immune cell population and their recruitment. So it's true that the osteoblast activity will be hampered by using this inhibitor, but the overall effect is the cell recruitment mediated effect, and it's only 15-day treatment. So I think we found a targeted effect in this way, like only inhibiting the Th1 cells or NK cells and thus seeing the effect.
Okay, good. Any other, particularly Swati, Shreyanko? Another thing, so when you have injected the melanoma cells directly into the tibia, right? So this is
Yes,
not a
yes,
bone metastasis model. Am I
yeah.
right?
So if you go through the paper, like there is two different model. One is intracardiac injection and one is intratibial injection. So for the like sake of presentation, I only presented the intratibial model data. We have also done the intracardiac injection model where we injected the cells directly into the left ventricle and then allow these cells to home into the like bone. Mostly we found that the cells are homed at femur, tibia, like long bones, and also on the skin, abdominal cavity, uterus for the intracardiac injection model. And we found a similar effect. We have done all these experiments with intracardiac tumor model also, and the effect is similar.
So you mean to say that when you are doing it through by intracardiac, when it is entering into other tissues,
Mm.
you get the same effect of microbiome with respect to its homing in, for example, uterus or non-osseous tissues.
We
Select,
didn't in
the selective
we
effect
didn't
for bone. Yeah.
we didn't investigate the effect in other non-osseous tissue like uterus or like the skin area. Like these are melanoma cells, so they go to the abdominal wall area. We didn't investigate the effect, but the major tumor site after the intracardiac injection is long bone. So we mostly focus all our study towards the bone and what happened inside the bone microenvironment. But it's true that cells are definitely going to the other organs, and the effect that you can see with intracardiac injection is maybe people can argue that it's a little non-specific effect. That's why we perform this targeted injection model, like the intratibial one, where tumor is only present within the tibia and no other part of the body. So that's why it's a targeted study, and that's why I only select this intratibial model data for my presentation to avoid any confusion.
I'm just curious that when you do intracardiac and when
Mm,
the home to bone,
mm.
what do you typically see? Because these are labeled cells. What do you typically find the most accumulation? In which part of the bone you find the major accumulation of these tumor cells?
Okay, so in terms of the particular area, the trabecular area is the most affected one. So the metaphysis area, you can say, and partial diaphysis. But the metaphysis, like both femur and tibia metaphysis area, is the most affected when you inject this cell. Like
Does
major
it?
tumor first
It
happen there.
doesn't. It doesn't go to, these cells don't go to a rib or in spine.
Uh, Ribs, yes, in ribs you can see some lesions, but we are not able to perform a microcity of ribs. But when we do a whole body X-ray, we can see the lesions. And spine, yes, there are some small lesions, but when we do the microcity, we didn't find any major tumor lesions as like compared to femur and tibia. And this is one difference that you can say like happens from human to mice. Like in human, the spine area is the most affected one. Like if a melanoma patient have metastasis, the cells definitely go to the spine and the long bones. But with mice, we found that the major target area is long bone. In spine, there is some lesion, but not as much as compared to the long bone.
No, that is fine. But with respect to the IV's data, that the cancer cell accumulation, the intensity, you always
Mm,
find it in the long bone. Forget about the lesion.
mm.
Is the accumulation intensity is more in the long bone compared to spine or any other bone regions?
Yes, so it's a limitation of IVs scanning. Like you, when you are using IVs to detect a cancer cells, you need a certain amount of luciferase activity to be detected by using IVs. So if the cell number is low, then it won't be detected with the IVs. And the second thing is we are using black 6 mice, and so due to those black hair that actually interfere with the detection. So it's reported that if people use any knockout mice with white hair or nude mice, particularly with nude mice, iris detection is not a major problem. Like there is no hair, so the interference is not there. You can easily, the detection capacity is much better. But with our case, it's a little limitation for our study that due to the presence of hair and that to like a black hair, you need a particular number of cells uh, present in our organ to be detected inside IVs. So as the long bone tumor volume is high, that's why they are detected inside the IVs. But when we do the necroscopy, we can see the lesions. And when we do the X-ray, whole body X-ray, you can see those minor lesions.
Wonderful. Let others ask question. Any clarification, please?
Hello,
Yes,
am I audible?
yes,
Uh, okay, as we know that immune system in each of us body actually respond to one particular disease in a very different manner, right?
yes.
So, can it happen that as we were talking about gut microbiome as a whole, so some
Mm.
species are responsible for this phenomenon, like they are promoting the migration of these NK cells and Th1 cells to a particular site, while the others are not.
Like in some human population, you are saying that
Yes.
it could promote the migration. In some human population, it won't. That's
Yeah, because some of us are susceptible to one particular disease,
yes,
and
and
why?
that's
Yeah.
That's true. So it is absolutely possible. It all depends on your gut microbiome composition. And like at the start of my presentation, I told that the gut microbiome actually is helpful to keep your immune system in better condition. We are currently doing those particular microbiome sequence analysis and seeing that particularly presence of which bacteria is responsible or like have beneficial effect on these NK and Th1 cells expansion. But it's definitely true. It has been seen in case of like glucocorticoid treatment case, like in some human, uh, the glucocorticoid response is much more detrimental compared to others, and the reason is the gut microbiome. uh, those humans have a different set of gut microbiome, like you can call it a nasty microbiome or bad microbiome. That's why their immune system are not acting properly. So it's absolutely possible. We are doing some study in mice, but we didn't finish it yet. That's why I can't show the result.
Yeah, fine. Thank you.
Yeah.
Okay. Is there any other question? Sorry, I had to leave briefly. If there is no further question, then please join me to thank Dr. Subhasis Pal. And again, I congratulate you and thank you very much for this presentation. Wish you
Yeah.
all the best
Thanks.
for your future.
Yeah. Thanks for listening my work. Thank you.
Bye.
And bye.